Okay, so hello everybody. Hopefully everyone has successfully moved into this room here. I am happy to introduce Dr. Sue-Ann Campbell to give our first research talk. This is sort of fun for me because Sue-Ann actually invited me to give my first ever talk at conference a couple summers ago at SciAm, so it's sort of coming full circle that I'm now able to introduce her. Sue-Ann got her undergrad at Waterloo, did her PhD in the States at Cornell, and is now actually back at Waterloo as a professor in the Department of Applied Mathematics. And her research is very much more on the mathematical side of things, being a professor of applied mathematics. And she's using these mathematical models to understand biological systems, including various types of oscillations in the brain. So with that, I will hand things off to Sue-Ann, and we will get off and running.
Great. I'm getting a lot of feedback.
Okay, first hiccup. Try talking again, see if the feedback.
Yeah, I'm trying.
Shoot. Okay, hold on. I'm going to close you out and then re-invite you up onto the screen really quickly. Hopefully that will fix this. Okay, we have our first technical problem of the day, or second actually. These are bound to happen.
Hey there.
Oh no. Okay, next thing to try. Jess, I'm going to close out again. Just hit refresh on your browser. And that has worked before. So hit refresh. I'll invite you back up. And hopefully that'll fix things. So again, my apologies, guys. This is one of the challenges of running a conference remotely, as I'm sure you are all aware. Hopefully this will fix things. So Sue-Ann, I just re-invited you up on screen. Hopefully that'll get things up and running. So Sue-Ann, did you get the invite up onto the screen? Why is it not letting you up? So I just resent it, Sue-Ann. Okay, accepted and connecting. Okay, say something.
No, I think I figured out what the problem was at my end. Sorry about that.
No worries. Okay, so once again, I'm happy to introduce Sue-Ann Campbell, who has broken the seal for us on these issues. So Now, no one else can be nervous anymore. We've fixed it. We're good to go. So Sue-Ann, if you can share your screen, and don't worry, take as much time as you need. We do have a coffee break sort of built in, so we have some leeway time. So I will focus in on your talk, and yeah, thank you for being here, Sue-Ann, and I'm excited to hear about time delays in microcircuits.
Okay, thank you so much for the invitation to speak in this meeting, and my apologies for the problem there. So yeah, I'm going to give a little bit of motivation for what I want to talk about, and then basically spend a little bit of time on time delay systems, and if there's time at the end, I'll look at a more application side of things. So the first one's a little more theoretical, and the second part a little more application. So so the motivation, so this is some old recording by John Milton, who's this is when he was at McGill University. So he's worked in epilepsy, and has a PhD in physics as well. And this was a recording of an adolescent with absence seizures. This was kind of a typical seizure at the top, and then at the bottom there was the thick noise during the recording, and you can see that the seizure actually terminated somewhat earlier. So I know Scott already talked about seizure initiation and termination, and there's different ways people think about this. And one of the, in this case, you might take this as an indication that perhaps this is a bistable system, that you can knock it out of this seizure just by doing some perturbation to the system. So so I find that kind of really interesting. And from the time delay, so that's kind of one thing that I'll be talking a little bit about, was sort of bistability, and Scott's given a really nice introduction, so that's great. I'm really interested in time delays, so here are just some schematic that I pulled off from somebody's nice webpage. If you have a cell here, and you have the axon, and you're going to the some other cell over here, then it takes some time for an action potential to travel from one neuron to another. So time for the information to propagate. The things that affect that, so that time delay, the axonal conduction time, the diameter of the axon affects it, how far it has to go, so how far apart the two neurons that are trying to communicate, and of course whether or not the axon is myelinated. So there's a really nice review on Scholarpedia by Swadlow and Waxman, who look at some ideas about this. This is looking at the fiber diameter and how the conduction velocity changes with fiber diameter myelinated versus non-myelinated. They also have great data showing what the kind of distances are and the corresponding velocities and the time delay. So the time delays can range from a few milliseconds to more than 100 milliseconds, depending on what parts of the brain you're thinking about connecting together. So I'm interested when they can be large and how that might affect the dynamics or the behavior that you see in the brain. Okay, so I'm going to give a little introduction to time delay systems, since I know there's a lot of trainees out there. So Scott already introduced the idea of differential equations. Time delay systems are generally modeled by delay differential equations. So in this kind of differential equation, the derivative of the state at the current time depends on the value of the state in the past. So a really simple example of this, so this is a really simple model, which you can think of, any engineering type people, comes from a feedback control type system, where you maybe are measuring something about your system, and then you want to use that measurement to apply some control to change the system. And there takes some time between when you measure the output and you apply the feedback. You have a differential equation that looks like this. So you have some inherent dynamics of the system, and then you have dynamics coming from this delayed feedback. And so this is a typical delayed differential equation. My derivative at the current time depends on the value in the past. If you think about a neural system, then you have many different neurons connected together, And so you can have, they have their own intrinsic dynamics. And then when you connect them together with time delays, you get a whole bunch of terms like that. So this is kind of a generic type of differential equation model that you might see if you're thinking about taking into account time delays in a neural model. And so, for instance, here you might have your equations of your Hodgkin-Huxley model for your individual neurons, and here you might have your model for whatever kind of synaptic dynamics that you have. Okay, so what can we say about, so a little bit about delay differential equations. Why are they, what makes them interesting mathematically, and how does that affect the behavior? So if we take our little scalar differential equation, and as Scott mentioned, and the idea of a fixed point, so this equation can have a fixed point as well. And one way we look at the stability of that fixed point is to linearize around that. So now we get a linear delay differential equation. And as you, if anybody's taken sort of, well, first year calculus or first set of differential equation courses, you know you can study the behavior of this linear system by the characteristic equation. The characteristic equation here looks like this. And what's interesting about this characteristic equation is because you have a delay, you get this exponential term. So what does that mean? It means that you have many, many eigenvalues. So mathematically, we like to talk about accountable infinity. So many eigenvalues, most of them are complex conjugate. You can say things, so you can't just solve for all the eigenvalues and write them all down, but you can actually study them. And you have a similar thing if you want to think about stability of periodic orbits. So what does that mean? It means if you're thinking about stability of your equilibrium point and potential bifurcations, as Scott talked about in his system, then there's many different eigenvalues that can potentially cause a change in stability. If you're thinking about, I'm not going to talk about today, but if you're thinking about noise affecting your system, there's many different modes that might be excited by noise because we have many different eigenvalues in the system. So even if you have just this one state variable, when we introduce this delay, we can get this many kinds of eigenvalues. So I want to give just to give a little brief idea of time delay. So this is sort of an example that everybody can maybe understand. This is a lovely schematic that I stole from somebody online. So the shower control problem. We all take our morning shower and we want to adjust the temperature to be what we want. And depending on how great your hot water system is and where your shower is compared to where your hot water is between when you turn the taps and when you feel the effect of that. So you're feeling the effect of the temperature, you adjust the taps, then that stabilize your equilibrium point at a constant temperature. But if you have a too long delay between when you make your adjustment and when you feel, you get an oscillation. So you can think of where it's too hot, I start turning up the cold, I keep turning up the cold, I keep turning up the cold, but oh, I overshoot, and it gets too cold, and now I start turning the hot. And you can see how this might lead to an oscillation between hot and cold. And most people solve that problem with a particular kind of control law called act and wait, where you adjust it and you wait to see what happens. So you sort of take into account that delay. So this is just an example of a very simple system, how a delay can cause an oscillation. From a mathematical standpoint, we look at that little example we saw before. And as I mentioned, we can think about the eigenvalues, and we can think about how these eigenvalues are the values of lambda that make this equation 0. A and B are parameters of the system, tau is the delay. If we think about when we can have a bifurcation, when you might have an eigenvalue of 0 real part, we can plot those up. Here I'm plotting it in the tau versus beta, and these lines correspond to places where I have eigenvalues of 0 real part. And what we can see here, and these numbers are telling me how many habits of my plane, which is my parameter space. So down here I have 0. That means my equilibrium, my fixed point is stable. And you can see as I increase the delay here, I cross a line and I get to a place where I have 2. And so that is one way that I can create oscillations. And I think Scott probably talked about a Hopf bifurcation. So that's one way you can have Hopf bifurcation. So a delay system, a delay can cause oscillations through a Hopf bifurcation because of the way the eigenvalues are distributed. And so I'll be thinking about that a fair amount. So what I want to think about in terms of some neural application sort of modeling, at least ideas. First, I want to think of a simple, what something you might call neural activity model. I'm just going to think about two different, so you can have multiple, and I'll think about two different populations, right? There's some basic decay in the activity. So the X-ray represents the activity. We have connections between the different populations. So there's some nonlinear connection between the population. There's some delay, time for the information to go from one population to the other. We also allow for self-feedback. So we're thinking of this as sort of a population model. And we also allow for delay in that and some. So this is kind of a nice cartoon model to play around a little bit with and see some things that can happen. We have our connection strengths, and those connection strengths are going to determine whether we have sort of excitatory or inhibitory interactions. And what I'm going to think about a little bit is, depending on whether we have excitatory or inhibitory interactions, what kind of things can the delays do? So the first thing that I want to mention is that delays can lead to long transient oscillations. So if we have two, and now I'm specifically thinking of two excitatory populations. So I'm going to show two examples. Here's one. This is going to be two excitatory populations. This is two inhibitory. You can see similar things. If this was an ODE model, so one thing you can show is you can get a situation where you have two stable equilibrium points and one unstable. So this is, again, exactly like a picture that Scott showed at the very beginning of his talk, where we had the unstable was at the top of the hill and the two stable guys were at the bottom of the hill. If it's an ODE system, this is two-dimensional, so I'm plotting it in the plane. I have equilibrium points. Any initial condition most initial conditions will go to one or the other of these equilibrium points. This is this bistability. We have this separation line here, and if I start really close to that separation line, I may take a long time before I head to one equilibrium point or another. And this is kind of what's showing here. You may sort of stick on something and then eventually you head. So this is versus time. I'm plotting one of the variables versus time. You'll tend to one of those equilibrium points. Now, what happens if you introduce a time delay into the system? It's essentially like adding a lot more dimensions. So instead of just being a two-dimensional system and I have this sort of line that I may follow along and then dimensions. So for instance, I could have directions where I'm oscillating towards that equilibrium point. So what you can have in time delay systems is you can spend, instead of following a line, you can spend a lot of time sort of oscillating close to this unstable equilibrium point and then gradually going to this other one. So these are some simulations of the, again, very simple system, just two variables with some time delay. And you can see how you can spend a lot of time oscillating before eventually going over to this one state or another. And this was first noticed and studied by Pachtemann in 1998. So this is an interesting from a point of view of maybe our system could be on a transient and we don't really care about where we end up at the end, right? So we can get these sort of very long transient oscillations when we have time delays. The other thing you can get besides just bistability, as is on the last example, between equilibrium points, you can get bistability between oscillatory cell and an excitatory cell. So this is common in this kind of setup, one inhibitory, one excitatory. This is a similar plot I had with my simple scalar model, where I'm plotting the eigenvalues with zero real part. And so what you can see here is that now, instead of before I had curves that were kind of nested, I can have curves that cross. So what this means is that I can end up essentially with more than one periodic solution. So if I cross here, I get one type of periodic solution. If I cross here, I get another. And so again, as I'm varying some delay, this is the sum of the delays in the loop here. Then I can create these oscillations and essentially with different frequencies. So I'm going to show you again some simulations And here what I'm doing is I'm following one of the oscillations, then we're giving a stimulation to the system. You can see it goes away for a while and then it comes back. If I change the timing of that stimulus a little bit, what we see is that we actually change to a different oscillation. So we've changed the frequency, and that's because there's a bistability. There's actually two oscillatory solutions. So a small different timing of when the stimulus happens can change from one to another. And also a different stimulus can also make us go to one of these transients. So in this case, I have a transient torus, really. So two frequency oscillations. And so quite the analogy of what we have with the equilibrium points. Now I can go away for a while and then eventually come back. So this is something else that in a really simple system you can see. Okay, so what about in more complicated systems? So in spiking neural models, so those were basically simple equilibrium systems. If there was no time delay connection, right, everything would just go to an equilibrium point. If you make more complicated dynamics at your nodes, such as Hodgkin-Huxley equation, then more complicated things can happen, but they're all in a very similar kind of vein to what we saw in those little examples. And I haven't attempted to, like, there's many, many papers, there's many ways, different ways that people approach studying this. So we see this idea of multi-stability as we saw in the simple model case, multi-stability between equilibrium points, between periodic solutions that come up through time delays. And a second thing that we see when we start looking at more complicated models is that you may affect a synchronization. So if you think of your individual neurons as oscillating on their own, and then we couple them together, and then we put time delays in that coupling, then that time delay can really affect the synchronization between the neurons. And again, there's a long history of people who've studied that in various different ways. One of the ways I like to study it is using the idea of bifurcation theory. And so I'm going to just present a little example about something that you may see. So in these more complicated systems. So again, this is, doesn't matter, it's a system of, I think, 6 Morse-Lacar neurons coupled together in a particular way. So obviously how they're coupled, the network structure is important to what happens. But again, what I'm plotting here, this is coupling strength versus time delay. So same as I've been showing in a lot of the other diagrams, is essentially delay versus coupling strength. Here, the system has, if there's no, there's places where you have stable equilibrium points, and then they can, when you cross these curves, again, what I'm plotting up is essentially these curves where you have pure imaginary, pairs of pure imaginary eigenvalues, and these can lead to Hopf bifurcations that can lead to oscillations. Why are these coded with different colors? Because in this system, the bifurcations that you get that lead to the oscillations have different synchronization properties. So red is that they're all synchronized together, and the other ones are various sort of cluster states where different ones. So I think blue is, it splits up into 2 groups, and they're a half a period out of phase. So you can see when we start to add more neurons, and we have these sort of inherently neurons, and where the inherent dynamics can be oscillations, then the delays, it's the same basic structure, right? We have to like get different kinds of oscillation, different kinds of synchronization properties that come out of that oscillation. And then here's a little example. This is, I think, 8 neurons, just to illustrate. So again, here we've got a system that's got stability between 2 different oscillation types. In this case, the difference in the oscillation type has to do with the synchronization of the neurons. So there's 8 neurons. I believe in the one, in this green one, they're all synchronized exactly together. So they're all oscillating on top of each other. So I tried to plot the voltages of the different neurons first in time here. In this other oscillation here, they're split into 4 groups of 2. So you can see the kind of different spikes are that kind of green, red, and yellow and beige or something. So those are the 4 different groups of 2. So
Joanne, I'm going to just jump in here real quick. maybe finish up within the next minute or so, just so we can get to some of the questions that the trainees have.
Yeah, sure. That's just great because I'm just about at the end of this part.
Perfect.
Yeah. So from here, just want to point out this is the synchro, so we can make a stimulation and basically jump from this one type of oscillation to the other. And if we look at the average of the voltage, then we see an interesting thing where when they're not synchronized, we get this kind of low level situation, and when they are synchronized, we see this kind of higher higher signal coming from the system. Okay, so I think what I'm going to do then is skip the last little bit about the absence epilepsy. Maybe I'll just skip to the end and show you the results. So the bottom line with, let me say, actually, I'll say one thing about our hypothesis. So absence epilepsy has to do with, it's associated with a particular neural circuit, which has to do with the thalamus and the cortex. And we had a hypothesis that the change in nerve conduction time in this thalamocortical loop, which happens as children age, so they get more myelination and the time delay becomes smaller, might account for the loss of seizures in adolescence. So a lot of children with this disease lose their seizures sort of spontaneously. So we built a little model for this. We put in some time delays. We looked at adjusting the time delay appropriately for aging, and we put in an adjustment of GABA conductance. Basically, this had to do with a particular genetic defect that's associated with it. And what we could show is that this is now the conductance, and this is the time delay. So the idea here is when they're young, the time delay is old, sort of large, there's bistability. We're sitting in this sort of genetic range of this conductance, but when they age, they move out of that bistability range. Even though the genetic defect is there, we don't see that behavior because we've lost the bistability because the time delay has changed. Okay, so I'll just go to my conclusions. The idea of bistability and time delays can be important in your system. We have a little model we did on absence epilepsy where we showed that. And let me just thank my collaborators and students. And I will go out of full screen mode.
So I will give you a virtual round of applause on behalf of everyone, Sue-Ann. Thank you very much, and thank you for pushing through with us at the beginning. So we have a few questions here that I'd like to get to. So the first one is, with regards to these delay-induced independent oscillations, are they specific to brain phenomena like seizures, or do you think that they're more related to specific brain areas, such as areas with long axonal transmissions?
Yeah, so I think that if they would be associated when you have two regions where there's a long axonal, it's a long delay in between them. So in the example at the end, that's what we were specifically looking at between thalamus and cortex. There's quite a distance there, and in fact, one way is quite fast and the other way is quite slow. Actually, it has to do with how they're myelinated. And so that was like why it was important in that situation. Yeah.
Great. So the next question sort of is a little bit similar. What is, I guess, more important is very subjective, but what do you think is more important when it comes to these models, implementing axonal delays or the synaptic dynamics?
So I like to have both. So if okay, yeah, because
that's the question. The answer I was expecting.
Synaptic dynamics do introduce a kind of delay of their own, right? Because it takes some time to process the information. So you can have that kind of delay as well as having a conduction delay. So some simple models, I mean, you don't bother with the synaptic dynamics. I put the delay in and kind of say, okay, that's going to take care of the synaptic delay. It doesn't take care of the details of the synaptic dynamics. But again, it depends on the system you're modeling, right? If you're looking at one particular brain region where the cells are pretty close together, then synaptic dynamics are going to be important. And the conduction delay, probably not so much, right? If you're thinking of two connected regions, then that's where they're kind of far apart, then that's where it's going to be important to include that conduction delay.
Awesome. All right, so next up, Dimitri asked, is there a way to distinguish, just looking blindly at the stimulated data, slow oscillating transients from delay from a conventional ODE limit cycle? So I think essentially what he's asking is, can you differentiate just by looking at something whether what you're seeing is actually a limit cycle or whether it's this sort of decaying oscillation to a fixed point?
Yeah, so that can be difficult. I'm trying to think if people do time series analysis, right? Especially if you're talking about data. It could, and that's why I pointed out in the example that you could kind of see the both. And then I kind of talked about bistability between oscillations, but like my collaborator, John Milton, is big on these transient oscillations. He said, maybe all seizures are just transient oscillations and we don't have to worry about limit cycles. And yeah, and I think with data, it might be hard, right? Because there's noise in the system, right? Anyways, that's perturbing you, even if you're on a limit cycle. So I don't know of a way. I don't know a lot about time series analysis. Some pretty smart people out there who do time series analysis. So there might be something I don't know about.
Okay. So yeah, so in basically just to sort of provide some context there for Dimitri's question, the idea being when you look at a system that is just sort of oscillating but decaying, if there's noise in the system that keeps perturbing that, it can end up sort of keeping the oscillation away from the decay. So it looks like just a sort of typical oscillation, but if you take the noise away, then it would just sort of go down to that stable fixed point. So when you're just looking at the data and you're not looking at the mathematical model, I think it's the answer is it's very difficult to determine whether it's a true limit cycle or whether it's some of these sort of noisy, perturbed oscillations. So let's do, I think, one more question here. So when you're adding more neurons, do you use all connections or do you use some random connectivity? And do you think that changing the connectivity scheme is going to affect your results?
Definitely the connectivity scheme affects it. So we've done, in terms of the analysis, we've done all-to-all, we've done what's called circulant coupling, which allows kind of like nearest neighbor and variable strength. So the kind of mathematical, well, some of the mathematical stuff that I've done, we need some sort of structured coupling, although there is work on if you know something about the eigenvalue spectrum, in the case of, if it's say random coupling, then you may be able to use that as well. But definitely that can affect the results for sure, because the network structure is really important in terms of these ones, especially the ones I showed where there was kind of different synchronization. Then how they're coupled together and how they're affecting each other is really important.
Awesome. So I'm going to give Sue Ann another round of applause. Thank you very much. So we are now going to end this session and we will move to the next one with Dr. Francis Skinner.